Whenever people listen about the salmonella, they think about the raw eggs and the poultry like the chickens and the undercooked meats, and they think about the food is free from the contaminations because salmonella is present in the sewage water. So, in this lecture, we will discuss all the things that are related to the salmonella. So, let's get started. First of all, we will look at the salmonella. We will see what salmonella looks like. So, important thing in the salmonella, it's a bacteria, Roche bacteria, gram negative. It's look pink color in the microscope. It has plasma membrane. Outside the plasma membrane, you will see the cell wall, and outside the cell wall, you will see the capsule. But some species don't have capsule, but majority have. Along with that, you will also see the presence of the pili that is used for the conjugations of the different bacteria. And after that, you will see the presence of the flagella that helps in the movement of the salmonella. So, three things in the structure that will act as antigens will be, first one is the cell wall. Cell wall will act as antigen. Most importantly, a polysaccharide component, it will be the O antigen. Next is the capsule that will also act as antigen, and that will be represented by the VI. VI means virulence, because those bacteria having capsule will not be easily ingested by the other macrophages. And third is the flagella. In the flagella, also act as antigen, and that will be represented by the H. So you will have three antigens, O, VI, and the H antigens. So you have to remember these antigens by heart. Next, culture properties. On which media we can grow salmonella? So most importantly, on the meconium, that is good for the gram-negative bacteria, and salmonella itself is a gram-negative bacteria. So it's a good media for the salmonella. It forms the colorless colonies because in the meconium you will see the presence of the lactose, and salmonella does not ferment lactose, so it will look colorless, while those species that ferment lactose will look pink in color. Next is the S as Salmonella shigella agar. In that case, you will also see the colorless colonies, but at the center, you will see the presence of black centers due to the productions of the hydrogen sulfide by the Salmonella. Next, xylose lysine deoxycholate agar, XLD agar, on which you will see presence of the red colonies, and the center will also be the black. And nutrient, in that case you will see the presence of the large and the moist colonies, and the nutrient mediate is mostly used for the sensitivity, antibiotic sensitivity testing. And motility, its motility test will be positive, as we have seen that they having flagella, and flagella gives them motility property. Next, if we look at biochemical test that is used for the differentiations of the different gram-negative bacteria. First is the catalase. It means that salmonella produce catalase enzyme, and it gives the catalase positive reaction. Methyl red will also be positive, and the citrate will also be positive. So these are the three most important tests: catalase, methyl red, and the citrate positive. Third is the TSI, triple sugar iron test. We take the test tube, and in the test tube we'll take the media. So in that case, we make the curve or slant like structure at the top, and at the bottom we will see the butt. Slant at the top, butt will be at the bottom. At the slant side, slant will be alkaline, and it gives red color, while the butt is acidic, and it will give the yellow color. So this is the important thing. And two most important thing which you have to remember, it will produce the hydrogen sulfide H2S, and H2S will produce the black color at the center, and along with that you will also see the bubble formation in the test tube that shows that it uh, produces the gas. Next, these are the identifications of the Salmonella different tests, and we will look at the types. Salmonella typhi, that causes the typhoid fever. Salmonella paratyphi, it has further category A, B, C, and it causes the paratyphoid fever. So these are the Salmonella typhi and paratyphi causes the typhoidal salmonellosis. Salmonellosis is actually all the infection caused by the Salmonella is called the salmonellosis. So in that case, Salmonella typhi and the paratyphi causes the Salmonella typhoidal salmonellosis. Next, Salmonella typhimurium and Salmonella enteritis causes the gastroenteritis. Gastro means stomach and enteritis means small intestine. This is the stomach. Food poisoning, in the food poisoning, our stomach and the small intestine also involved. In that case, stomach plus small intestine. And enterocolitis, entero means small intestine, colitis means large intestine. So these different names are the diseases caused by the typhimurium and the enteritidis species, but you will see the we will categorize it according to the location of the gut. Gastroenteritis, stomach and the small intestine. Food poisoning, location will be the stomach and small intestine. And enterocolitis, entero means small intestine and colitis means large intestine. Salmonella colerasius, that causes the septicemia, in which you will see this species goes into the blood and multiply and produce the toxin, mainly endotoxin, because it's a gram-negative bacteria. And ultimately, this infection goes towards the other organs. If it go towards the bone, it is called osteomyelitis. That is the most common seen in the sickle cell patients and the cancer patients and those children affected by the enterocolitis. And if it move towards the lung, it causes the pneumonia, and the if it go toward the meninges, it causes the meningitis. So from the septicemia, you will see the bacteria spreads to the in the body and goes towards the different organs. Now this category that are not related to the typhoid is called non-typhoidal salmonellosis. So that is the important thing. So these are the different types. An important thing which you have to remember about the Salmonella typhi, this species mostly occurs in the carrier state because it multiply in the gallbladder, and it is mostly common in the woman. It is common in women and it multiply in the gallbladder. So carrier state mostly excretes the Salmonella typhi in the feces, and feces can contaminate our food and the water, and if someone drink or eat these food or water will be affected by the Salmonella typhi. So your food and the water must be clean if you take it at outside the home, because mostly in the restaurant proper care are not taken, and in that case the family are mostly affected by the Salmonella typhi due to the proper unhygiene food. So that is the important thing which you have to remember. Pathogenesis, how this virus entered in our body. So it entered through the contaminated food and the waters and the raw eggs and the poultry like chickens or undercooked meat. So lot of different sources, and it entered in our stomach, but our stomach produce the HCl, but it can prevent from the HCl and then enter into the small intestine. So our main is the stomach component of the GIT. Second is the small intestine, and the large is and last is the large intestine. So these are the major locations where Salmonella can attack: stomach, small intestine, and the large intestine. So after that it will enter in the enteric cases, like in the you will see the entire gastroenteritis, food poisoning, and enterocolitis, this will remain in our gut. It will not be spread into the blood, but in majority cases. But in the typhoid fever, it can enter from the small intestine into the bloodstream, and in that case you will see the bacteremia, presence of bacteria in the blood, and after that you will see the different symptoms related to the typhoid fever. But the most important species that present in the blood and replicate or multiply itself and produce the toxin is the choleraceus, that causes the septicemia, and ultimately it can also go towards the septic shock, where low blood pressures and the hypoperfusion of the different organs happens in the septic shock. So this is how salmonella entered in our body and caused disease according to their location, mostly related to the gut and related to the blood stream. Next, we will see the symptom in the typhoid fever. You will see the high-grade fever, weak rose spots on the abdomen, and constipation at the early, and diarrhea. That is the rare case, diarrhea at the later stage. But mostly you will see the constipation, hepatosplenomegaly, and the tender abdomen, like pain when touching the abdomen. These are the symptoms related to the typhoid fever, and the paratyphoid fever will be mostly similar, but will be mild in that case. Next is the enterocolitis, food poisoning, and gastroenteritis. These symptoms will be related to our gut, GIT, like nausea, vomiting, abdominal cramps, and the diarrhea. But important thing, in the enterocolitis, you will see the bloody or mucus diarrhea. You will see the mucoid diarrhea in just enterocolitis, while other symptoms will be mostly similar in all the cases. Next is the septicemia. Septicemia means bacteria replicates in our blood and produce the toxin and causes the symptoms like the high grade fever, chills, and ultimately it can go towards the septic shock, where low blood pressures, and you will see the hypoperfusion, means that our organs will not get proper oxygen in the septic shock. In that case, lab diagnosis, we will go toward the culture. We will take the blood in case of typhoid, because in the typhoid bacteria enter into the blood, so in mostly 1 to 2 weeks of the illness. Next, we can take the stool and the urine samples after the first week and the second week. But in the first week we will take the blood. But important thing, we should not take antibiotic before giving the sample. So you have to take the blood sample, and after that you can go towards the, after the results you can take the antibiotics according to the antibiotic testing, sensitivity testing. Withdrawal test, in which you will see the detection of the antibodies against the O and the H antigen. O for cell wall, H for flagellar. PCR, we will take the blood and the stool sample and we will detect the DNA of the bacteria. Rapid test, you will see detections of the antigens. Three most important antigens mainly we have categorized. In the treatment, antibiotics, ciprofloxacin, azithromycin, ceftriaxone. But in the cases like related to the gut, we will normally do not take antibiotics, and it's just a self-limiting, but we will take the other management like the fluid replacement to prevent the dehydration in case of diarrhea. And in the prevention, you will see the two vaccine, typhim VI, in which you will see the virulence polysaccharide, and the typhoid 21A, in which you will see the live attenuated bacteria. So typhim VI is taken through the intramuscular route, while the type 21A is taken through the oral route. So these are the vaccine related to the typhoid fever, not related to the other gastroenteritis condition. So this is all about the salmonella. If you still have any question regarding any point relating to the salmonella, its species, its properties, its test, you can ask in the comment section. Thank you so much.